Well, animals have evolved to occupy every niche on the planet where we would ever want to operate a robot. As such, they are proven solutions to the problems of navigation, obstacle negotiation, and investigation. So the goal of my research is to find ways to build robots that capture the capabilities that animals have. So we do this at the Marine Science Center in Nahant, Massachusetts. And my background is actually in neurophysiology and a branch of neurophysiology called neuroethology. And what we try to do is to explain the behavior of organisms in terms of the properties and connectivity of networks of nerve cells. We try to understand the dynamics of how their synaptic networks operate, how they respond to contingencies of the environment, and also how we can achieve engineered devices that have the capabilities that these animals have in the natural environment. We also eat the experiments. Now, the paradigm that I was trained in is what's called the invertebrate central pattern generator paradigm. And this is a little mini brain in the lobster that controls the movements of what Julia Child used to call the head sac. And the head sac of a lobster is a combination stomach, sort of a gizzard, and a small intestine in one little package. And this network controls its operation. Each of these round things you see here are the cell bodies of individual neurons. And we can insert micropipettes into them and record their activity and identify each of them uniquely. When you can identify all the neurons and record from pairs of them, you can establish their electrical interactions, which are called synaptic connections, figure out how all the neurons interact. And when you look at all possible pairs of 30 neurons, this is the network that exists in this ganglion, which we know in exquisite detail. We can also record the transmembrane voltages of the cells, inject current, look at how this perturbs the rhythms that they generate, and also look at pharmacological manipulations to see how various drugs alter the way these operate. The end result of this is a model which is called the command neuron coordinating neuron central pattern generator model. And this holds that each limb is controlled by a network of neurons. It's called a central pattern generator. And the central pattern generator can produce, for example, walking in the absence of sensory input from the periphery and also from descending information coming from the brain. We have one of these in our lumbar enlargement. We have one in our cervical enlargement that control the movements of our legs and our arms. Now, in multi-limbed animals, there's a central pattern generator for each body segment, and they're coordinated among themselves by a category of neurons called coordinating neurons, which send information from a governing oscillator to a governed oscillator that maintain the interleg gait. And the role of the interleg gait is to maintain a pattern of support against gravity to stabilize the organism in the pitch and the roll plane. The final thing are neurons called command neurons. And in lobsters and crayfish, if you take the brain connectives going from the brain down to the rest of the nervous system, you can fray them into little fibers, each of which is a single axon. And if you stimulate these axons electrically, you can initiate, for example, forwards walking, backwards walking, swimming, feeding, pretty much everything you see. And there's a one-for-one correspondence between these neurons and recognizable behavioral acts. Now, this model is generalizable, and if you reorganize these components, you can produce a central pattern generator for a swimming organism. So we've used this architecture as a basis of a process we call biomimesis to generate biomimetic robots. So this is the first time we ran our Lobster robot. So we got it right. This is a movie I'm so proud of. And these robots are quite unique. They have a biomorphic plant, meaning they have the shape of the animal because the shape of the animal gives the animal biomechanical advantages in the environment. They have a neural circuit-based controller. They have sensors that code in the same way as animal sensors. They have artificial muscle. And then the robot has a library of behaviors that we reverse engineered from studying the behaviors of the model animals. Now, this is the other model animal we use, which is called a sea lamprey. This is up in the Merrimack River, and this is our lamprey-based robot. Now, when we got to this point in this research, we realized a fundamental conundrum. if you try to control a robot with a computer program, unless you've anticipated every possible contingency that the robot's going to experience and have a programmed a detailed escape strategy, it's going to get stuck. Pretty much all robots that exist right now are teleoperated. People call them robots, but there's somebody with a joystick steering them, okay? We're trying to make robots that can behave on their own. And what animals do when they get stuck is they wiggle and they squirm. And we're convinced that what the animals are doing is they're increasing the level of chaos in the networks that would otherwise generate locomotion and navigation. And the chaotic variations on the locomotion are the wiggling and squirming. So we want to build robots that can wiggle and squirm like animals do. And our solution is to build electronic nervous systems, but these nervous systems have to be able to operate in real time. So to achieve this, we've gone to some phenomenological models of neurons. And this is the model that we're using the most now. This was developed by a mathematician named Nikolai Rulkov. And this is what's called a map. And this is the membrane voltage in cycle n plotted versus the membrane voltage in cycle n+1. And there are two variables, x and y. Normally the thing is cycling around this loop, which gives these action potentials. And periodically it drops down into this low area, which is this slow component. And if you vary two control parameters in the equation set, which are called alpha and sigma, in this region they produce bursts of spikes. In this region they produce tonic firing. And in this area they produce chaotic activity. So we can assemble networks of these to run on a digital signal processor, and that forms our electronic nervous system. We don't program these with a programming language. We program them with the logic of neuronal connectivity and the dynamics of neurons. The other feature about this is that the development environment we use, which is called LabVIEW, is also the development environment for Lego robots. So we're working with Chris Rogers, the guy that wrote the software for Lego Mindstorms, to produce a neuronal module to add into Lego Mindstorms so that high school kids can program robots with brains. Okay, now these control signals, which are trains of pulses, don't make very much sense for controlling the normal actuators used in robots, which are motors. So we use an artificial muscle. And this is the length of a wire of a material called nitinol, which is a 50-50 alloy of nickel and titanium. And it's the material that they make heart stents out of. And it exhibits something called a shape memory effect, which means if you cool it or heat it to a very high temperature, it goes into a dense crystalline state. And then if you cool it down, which we use seawater to do the cooling, you can then stretch it out by about 5% over its austenite length. And then if you apply current to it, it will contract, and a 250 micron wire will lift a kilogram in about 150 milliseconds. So to make graded contractions, we use what's called the size principle of motor neuron recruitment, and we mimic small, medium, and large neurons with pulse widths of different duty cycle. And this is the upper displacement of a 1 kilogram weight. And you can see by varying the pulse width duty cycle, we can vary both the velocity of the contraction as well as its amplitude. So this stuff is like real muscle. So we build brains to control the individual central pattern generators. And this will give you an example of one of the sorts of networks that we use. These are the individual central pattern generators of each body segment of this lamprey-based robot. And this is the pattern of activity propagating from nose to tail that would underlie forward swimming. And so each of these circles here represents a neuron, and these arrows here represent the synaptic connections. And we can actually build these connections up. And this robot here is the first robot ever built that has a nervous system of neurons and synapses. And it has about 400 neurons and synapses in it. If we bias the excitation towards the front end of the animal, we get forward swimming. And if we bias it from the rear, we get rear to front swimming. So this gives us the ability to switch between behaviors, but it's changing the pattern of bias in the two ends of the animal. Now this is this thing swimming, and it has what's called a short baseline array. So these are 3 sonar microphones that can enable this thing to home on a sonar beacon. Now my sponsors, which are the Department of Defense, are interested in using these things to neutralize underwater mines. And nowadays the way they find mines is they have dolphins that swim down, find the mine using their biosonar, and they release a bite plate, which has a weight and a little balloon that floats to the surface. And then they send out a Navy SEAL with a satchel charge. He finds the balloon, swims down the string, and puts a bomb on the mine to blow it up. It's called detonate in place, and the opposite strategy is pick up and carry away, which you don't want to be doing with a mine. Anyway, this will enable us to have the dolphin instead drop a sonar beacon, and then this thing would swim and home on the sonar beacon. Now, I've shown you the central pattern generators, and there are these descending commands for backward swimming, slow and fast forward swimming, and amplitude recruitment on each side of the animal. And then we have a network of interneurons and sensors which mediate various exteroceptive reflexes to control the behavior of the robot. So this is one for heading control, which is activated by a compass that's bilaterally symmetrical. This is the input from the short baseline array. And then we have some other neurons which respond to accelerometers that detect impediment. So if the thing swims into seaweed or hits a rock, these trigger reflexes that cause the robot to adapt. So for the purposes of homing on a sonar beacon through impediment, this is the entire circuitry that we need in the brain. And we're in the process of implementing this right now. Now, the beauty of this architecture is that there's a very small part count. We can put the whole nervous system on a digital signal processor chip. It costs about $8. And then the interface to the actuators is just a 2-chip interface for each actuator. And we have a similar simple interface for the sensors. And the bottom line is that we can produce cheap robots. Okay, now what can you do with cheap robots? Well, we're working on a project right now to make a system for mine clearance. And this would consist of a radio and GPS controlled boat that would have a winch on it and a little well that we could launch robots out of. And it would have a grasper, which is a robot that would send out a sonar signal to draw in the robots and then pick them up and bring them back into the boat. And then we would put in this a set of marsupials, which would consist of bottom crawlers, swimmers, and a robot we base on the abalone we call Robalone. And in use, these would all be communicating with each other using sonar, so they would function as a unit and be able to communicate with an operator through satellite communications. And what we're really trying to achieve is autonomous behavior. And the kind of autonomy that we're talking about is what we call supervised reactive autonomy. And the idea is the same as if you took your dog for a walk and you threw a stick out into a pond. The dog autonomously is swimming out and retrieving the stick and coming back, and you're giving it supervisory commands. Now that makes the robots a force multiplier because you could be throwing a lot of sticks. So the idea then is that we would set out a series of Robalone to form what's called a long baseline array, and the robots would be given a mission, a search vector, which has a compass heading, a duration of time to walk on that heading, and a propensity to either negotiate or investigate objects that it collides with. Okay, it would then announce its position, receive a new search vector, and go off and do a search. Now, we also want our robots to exhibit great group behavior where they interact. And there's many examples in the animal kingdom of animals interacting. This is cueing behavior in spiny lobsters. But the other thing that arthropods bring to the table is a phenomenon called stigmergy. These are the nests of small, intermediate, and large complexity of a fire ant. They have this incredibly similar architecture, but the thing that's crazy about this is there's no architect and there's no plan. So the construction of these nests, which are incredibly species specific, results from low-level interactions from the individuals. And they do some crazy things, like lobsters communicate by peeing on each other. So these all form what we call exteroceptive reflexes, where there's a sensor and a reflex pathway like I showed you for the lamprey. Now, the great example for stigmergy is social insects. And so we also have a project to build robotic bees. And this is a flight actuator for a robotic bee. The wingspan here is about 1.5 centimeters, and here it's actually flying. And this thing can take off. So you'll see here, it now, supported by some guide rails, it will take off and bear its own weight. Okay, now the real bees have some sensors. They have what are called ocelli and eyes, a mini brain that weighs about 1 milligram. And we've divided this into a bunch of equivalent electronic modules. For example, these are the flow sensors in the eyes. These are ocelli for using UV to detect flowers. And I get 55 milligrams out of the 500 milligrams to build the brain in. So we're doing similar exteroceptive reflexes in RoboB to enable it to fly. Now, my most recent project is to build what's called a biohybrid robot. And our real goal in this is to give these robots a sense of smell. So we're using a paradigm called synthetic biology, where we start off with a chassis, which here is yeast. And then we insert into the yeast some genes for some receptors, a G protein, a calcium channel, and a reporter called aquarin, which when it binds calcium gives off a photon of light. And it turns out that at MIT there's a thing called the Registry of Standard Biological Parts, and you go there and they have a catalog of reporters, and you can pick one out, and there's a button there that you press, and they send you this spec sheet, and it has some little red dots in the bottom, which are the DNA. So you then insert those into your chassis, and you end up with what we call in synthetic biology a device, a chemoreceptor. And it turns out there's a receptor for RDX, which is C4. So this would enable you to identify a mind, for example. We can use an opsin as a receptor and construct a photoreceptor. And so what we're doing is assembling this now into a robot where we have synthetic sensors, an electronic nervous system, and then muscle in which we put a gene called channel rhodopsin. Now, this is the electronic nervous system. These are all motor neurons which will activate these muscles. And the way that we communicate with the muscles is we take the action potentials, use them to turn on an organic LED, which puts blue light onto channel rhodopsin, which is a gene we're putting in the muscle, which will turn on the muscle contractions. Now this thing's supposed to be 1 centimeter long, so fabbing it's a problem. So we're going to what's called inkjet printing. And it turns out they make printers that they print the labels on your DVDs with, and these can produce lines that are 50 microns wide. So what we're doing is taking the body axis, this would be the tail of the robot, and we plate some anodes on it with a resist, which we then etch away around the gold around the resist to produce the conductors. We then put on the polymer layers of the OLED, and then we print a cathode over that, and then we coat this with a muscle repellent, and where we want muscle to grow, we coat it with a glue, a molecule which will attract and bind muscle, and then we dip this into muscle, and we have a full body axis with all the control electronics and the muscles themselves. Okay, I just want to mention one last thing. We also want to build underwater robots for mariculture. Now the reason for this is nobody wants a fish farm like this in front of their waterfront property. They're major navigational hazards. Thirdly, they're subject to being broken up by storms and losing the stock. And finally, they're quite polluting. So we have a robotic solution to this where we make a big tank about 60 feet in diameter, about 15 feet deep, and that will have a 3-stage biofilter system that takes out solid waste, nitrates, and phosphates, be controlled by an electronic nervous system, and in operation we would pull this underwater so it's no longer an eyesore, it's not a navigational hazard. We would use wave energy to power it, and when we get into big storms we would just pull the thing deeper underwater. And it turns out wave energy falls off with a 3rd exponential of depth. So you can, at 50 feet, you can ride out a hurricane. And then to control it robotically, we're going to use underwater sonar and Wi-Fi communication so we can actually control this thing with an iPhone. So we call this approach neurotechnology. And we argue it has the same relationship to neuroscience that biotechnology has to molecular biology.